My name is Jonathan. Today I will be doing my presentation on the RNA world hypothesis, which is the idea that prior to the existence of life, prebiotic chemistry was centered around RNA and RNA-like molecules. So, just to give you some background here, the Earth is 4.5 billion years old. You probably remember learning that at some time in middle school or high school. Water itself didn't actually appear on the Earth for about from 4.2 to 3.8 billion years ago. It's extraplanetary in origin, meaning that it came from somewhere else. The geological evidence suggests that actually another planet that was full of water slammed into the Earth and basically introduced water to the surface, and coincidentally also created the moon. Now, interestingly enough, the earliest evidence of life points back to about 3.8 billion years ago. So either right around the time water arrived on Earth or shortly thereafter, life first emerged. And this organism is known as the last universal common ancestor. There's no fossil. However, they're just based off of what we know from genetics as well as some of the geological evidence, we can sort of predict when it first emerged. But that's not what we're concerned with today. What we're actually concerned with is what happened before the last universal common ancestor emerged. What were the steps that occurred? If you see, this is just a timeline of life. If you look at the very, very bottom, which represents the very, very first formation of the Earth, life emerged pretty shortly thereafter. And it's this little tiny gap between the arrival of the earliest water and the universal common ancestor that we're going to be talking about today. I chose this topic because it really represents a fundamental question that not only is scientific in nature, but is also philosophical as well. I like to think of it as the proverbial chicken and the egg question. With the question being, what were the steps that, what came first in life? Was it DNA? Was it RNA? Was it proteins, right? This is something that has been a concern of researchers for a very long time. And you learn the central dogma, right? In order to have proteins, you need to have an RNA, and in order to have RNA, you need to have DNA. But this doesn't make sense when you're going back to the original chemistry that would have existed, because as we know, DNA is a very stable molecule, which is great, but its single function is to store DNA. It does not catalyze reactions. It does not make proteins, right? It is unzipped, read, and closed back up. Similarly, proteins don't make proteins. Proteins don't make RNA, nor do they make DNA. RNA does all of that, and we're going to go into that, right? So how this is relevant to the course, it's got the central dogma, the catalytic abilities of RNA, plus the loop structures. These are all intrinsic properties of RNA that are going to be really important as we go through our discussion today. The paper I'm using, this was published this year, actually, I think it was back in August. It's a narrative of the RNA story, so to speak. These authors, Fine and Perlman, they posit the idea that RNA evolved before coded proteins in DNA genomes, that translation actually evolved before transcription and DNA replication, that the origin of life was a gradual chemical process that resulted in the birth of Luca. And their goal with this paper was to propose a plausible timeline of the RNA world hypothesis. Now, before we go into this, I just want to pull out this beautiful quote from the paper, which really sort of represents how this whole paper was written. Yes, it was very scientific in nature, but it also it's just a fantastic read. It's very interesting and quite easy to read as well. Life is the result of a particular arrangements of matter, and at no single point was inanimate matter transformed into a vital substance. Everything we're going to learn today emerged gradually. It's not necessarily the truth that these experiments we're talking about represent what actually happened. This is just an exploration of different pathways that could have taken place, and they used logic essentially to deduce what might have happened based off of these experiments. This is the single figure from the entire review. It is their timeline. It's divided into 4 stages. We have the pre-RNA world, the early RNA world, the late RNA world, and we have the near LUCA stage. All of these stars here are basically the topics that we've discussed in this class that you should be familiar with. But you can see basically how they emerged. We started with pre-RNA monomer formation, right? And then shortly afterwards, they formed into oligomers. As we go on through here into the early RNA world, we had the emergence of ribozymes, which are basically oligonucleotides that are capable of catalyzing reactions. And it gets, of course, as you can see, more and more complex. These ribozymes evolve until we start to get towards the very end, we start to see the emergence of proteins that are coded for by RNA. We're not going to go into all of this detail because it would just take too long, but I highly recommend reading the paper. So, the origin of life research really began back in 1953 with this experiment called the Miller-Urey primordial soup experiment, where they just basically proved that amino acids can form abiotically. That means without a template. They basically made a soup of methane, ammonia, water, and hydrogen, and boiled it for a couple of days, and then saw what came out of it, and they got amino acids. This has been criticized because that's not actually how it happened. That's not the actual content of the Earth's atmosphere at the time that this would have happened. But it's been verified over the decades through different experiments that simulate the conditions we know actually did exist. This experiment introduced the concept of prebiotic plausibility, which is just basically saying this is one way this could have happened. This is the chemistry that could have happened. And if it's not this chemistry, then it would have been a similar chemistry. This is actually from that very paper, just to prove that they did find some of these amino acids, so aspartic acid, glycine, alanine, and, well, a couple others that we don't really see as essential amino acids, but are amino acids nonetheless. So RNA would have existed as a monomer at first. It probably didn't exist even as RNA, but rather an RNA-like polymer. We have lots of examples that have been created. I have just included a few of them here on the screen for you. How they would have actually condensed to form longer, chains of RNA molecules, they would have needed some sort of form of energy. So it's likely that hydrothermal vents, hot springs, UV radiation, all of these are potential sources. I'm not going to go into this next part, but fatty acids and phospholipid membranes could have sheltered these autocatalyzing RNAs. This is something that is brought up and actually discussed in the paper, but it's a little bit out of the scope of what I'm trying to do today. The real problem, though, is that the phosphodiester bond that connects these monomers is energetically unfavorable. It's about 22 kilojoules per, to try and form that bond. But we actually know that minerals that have been dissolved in an aqueous solution are capable of actually catalyzing this reaction. This is not in the review, but this is actually an important paper to understand. It just shows that you can have as many, chains of as long as 5 monomers being connected together, from different clays and sulfides that would have existed on the early Earth. So we know that you didn't need an enzyme to catalyze this reaction. And that's important. There were no enzymes at this time. How did we get RNA to copy itself and to continue to grow? Well, this experiment was cited in the review. And all they did was take a solution of tetramers, basically of ribonucleotides, and enhance the nucleophilicity of their 3' hydroxyl end. And this basically made them ligate. This is all prebiotically plausible. By increasing the concentration of the tetramer and the catalyst, again, not an enzyme, just a molecule that is bound to a small metal ion, very common in nature, you saw an increase in the ligation rate. This is how you could get short ribonucleotides to copy themselves, not polyribonucleotides. If this happened, then how did ribozymes come about? Because ribozymes are polyribonucleotides. Well, it turns out there's this other experiment that's really cool to read about, very complicated, and I'm going to have to do a really short summary of it, but essentially, this process of loop closing ligation allows you to take a bunch of segments of RNAs that are, oligonucleotides in their length, and then they can be ligated together in order to form functional ribozymes, all without enzymes. Again, look at that word at the very bottom, prebiotically plausible. This is something that could have potentially happened on the early Earth. And the principle was simple. Take a ribozyme, reverse engineer it to its component parts, and then see if those parts can be reassembled through this process. And that's exactly what happened. They actually proved it by building a hammerhead ribozyme. Why a hammerhead ribozyme? It's because hammerhead ribozymes catalyze ribonucleotide ligation and cleavage. This is how you get those longer polynucleotides to form and make functional ribozymes that can further evolve and specialize without a genetic template. So just in terms of the timeline, now you have RNA molecules that can actually do things, that can catalyze reactions that sort of have a direction, right? And this is really important. We've been learning all about how proteins are made, we learn about the ribosome as made up of these protein subunits, but those subunits do not actually carry out catalysis of peptide bond formation. That is actually carried out by this RNA core called the peptidyltransferase center. It is ribosomal RNA. And this is a highly conserved sequence. In fact, the researchers even say that this RNA sequence might be from before Luca, meaning before life emerged on Earth, which I think is just totally cool. And it also shows that evolution works at the chemical level, not just at the biological level. And the paper goes on. It says that, once you had these complex RNA structures, things just, kind of went on from there. DNA would have evolved alongside RNA, probably as an RNA-DNA hybrid, and then over time they would have separated into DNA being sort of the template that RNA copies itself from. Eventually, as this got more and more complex, proteins would have emerged and become more complicated themselves, and thus taken over the functions carried out by RNA in this form of evolutionary specialization. Now, the question too is polymerases. This is something we've really been focusing on in this class, and in the very last few chapters, we learned about RNA-dependent RNA polymerases, which these are polymerases that do not need proteins necessarily to catalyze these reactions, right? The very first RDRP was a ribozyme, as far as we can tell. This is very similar to reverse transcriptase, actually. It is an RNA-dependent RNA polymerase. And there's a lot of connections to why RNA might be the center of this world at the time. DNA, in order to be replicated, needs an RNA primer, right? This dependency suggests that DNA couldn't have arisen without RNA first, right? So it seems to be, it went from RNA to RNA DNA to DNA. To finish off, just talking about this RNA world as if it's a past thing is really not true. It's still present today. We see it in every living thing. It is literally in the center of the central dogma. It connects the DNA to the proteins. Now, this paper did not talk about metabolism. That is a totally separate, realm of research here. And it is not a complete origin of life timeline either. Again, this is just to show a prebiotically plausible way this might have happened. It is very general in scope. We will never know the answers to this, which makes it kind of an interesting field of study. To me, this has a lot of implications about life in general, not just life as it is, but life as it could be. If you look at this diagram that I made, we took the RNA world, but we could have taken the TNA world or the PNA world, right? There's so many possibilities of these nucleic acid polymers that could be the basis for life on other planets, on different moons, right? And that brings me to, moving into the future here. We don't need to look very far into space in order to find organic molecules and amino acids. We find them on the moons and planets in our own solar system. There's a few environments nearby that are also, I guess, prebiotically plausible. There's that term again. Europa has actually a subsurface ocean that might provide an ideal environment for biogenesis. We have observed water plumes coming off of this moon of Jupiter. So life could be underneath there, and if not life, maybe maybe the chemistry for life. And it's not just Europa. There are other moons as well. Ganymede and Callisto both are also moons of Jupiter. They might also have subsurface aqueous environments. Saturn's moon Titan is too cold to have water, but it does have liquid methane. So you might not have, ribonucleic, or you might not have nucleic acids, but you could have something similar to nucleic acids there. I also encourage you to look into the clouds of Venus. This has been a hot topic in astrobiology recently because yes, the surface of Venus, terrible environment. Life cannot evolve there. But in the clouds, it's actually pretty peaceful and stable. So look this up if you're interested. But it's just fascinating to me to think of all the different ways life could have evolved. We only have one sample right now, but if we were to get another sample of life, well, that would be huge. We would learn a lot more about how life works. I leave you with this discussion question, coming back to our evolutionary principles. Think back to freshman year biology. Survival of the fittest is used to describe how organisms with adaptations most suited to their environment will survive long enough to pass on their genes to the next generation. This is why we find heat-resistant microbes inside hydrothermal vents and Arctic tuna with an antifreeze protein in their blood. How might this principle be applied to a chemical system? What properties of RNA or RNA-DNA hybrids might give them advantages in a chemical system? My advice for this question, think of RNA and DNA as organisms and go from there. With that, that's the end of the presentation. Good luck on your exams and congratulations on making it through biochemistry.